Hi, welcome everyone to our panel on Refsum disease research. I'm Kristie DeMarco, and I'm the founder and president of Global DARE Foundation, and I'll be your moderator today. I'm honored to be here with Dr. Joe Hacia and Dr. Nancy Braverman. They're going to introduce themselves in just a minute. But before we get started, I'd like to share a little bit about Global DARE Foundation. Global DARE Foundation was established in October 2019, and DARE stands for Defeat Adult Refsum Everywhere. Our mission is to promote worldwide awareness and better quality of life for all who are diagnosed with Refsum disease. Our goal is to support research, education initiatives, awareness activities like we're doing today at DareFest, and advocacy. Driving research is at the center of what we do because we dare to believe in a cure for adult Refsum disease. Dr. Hacia and Dr. Braverman are supporting Global DARE's mission by being valuable members of our Medical and Scientific Advisory Board. With our Medical and Scientific Advisory Board, we are raising awareness for Refsum, driving better treatment and care, and reinvigorating the research into better therapies for Refsum disease. Now onto a few housekeeping details. So you're obviously in listen-only mode. You don't have your camera live, but we do want you to participate with us. So you do have the ability to answer, to put in some questions. What you need to do is go to the right-hand panel, and you'll see a tab called Stage, and then you'll see a tab called Q&A. So just put any questions that you have into the Q&A section there. And today's session will be recorded for later viewing at the Global DARE website and on our YouTube channel. So now on to the fun stuff. So Dr. Braverman and Dr. Hacia, I'd like to have you each give an opportunity to give you your background and experience. So Dr. Braverman, if you want to get started.
Yeah, sure. I've been in the field of peroxisome disorders since the mid-1990s. I started during my genetics fellowship where I trained at Johns Hopkins, and there was a big group of researchers there in peroxisome disorders. And during that time, we identified several genes that cause peroxisome assembly disorders, which is one group of disorders. The ABCD1 gene for X-linked adrenoleukodystrophy was identified around the same time. And as well, the group that I was working with also identified the Phi-H gene, in which defects cause adult Refsum disease. So it was a really exciting time, I stayed with the field and opened up my own lab and generated mouse models and set up cell screens to look for drug therapies. And I've continued to work now on therapeutic development in cell and mouse models. And we also have set up a large natural history study in which we collect medical records and speak with families and follow them over time. And we started this around 2012, and we now have about 250 patients with peroxisome assembly disorders, which is quite rare as well. So that's a lot of patients in one study, and with that information, we've been able to extract medical data and help to inform different systems like growth and neurological progression and vision progression and hearing, which are some of the issues in that group of patients. I started in the early part of my work. I was working mainly with PEC7, which is a disorder in which there is accumulation of Phytanic acid in patients. And we were also aware of much milder patients which didn't have the RCDP phenotype, but had more phenotype equivalent to adult Refsums. And that was written up by my colleagues about a decade ago. And we recently pulled together more of these patients. And so with very mild mutations in PEC7, a peroxisome assembly factor, you could have a disease that looks exactly like adult Refsum because the only biochemical abnormality that's significant is the accumulation of Phytanic acid over time. And we recently published a mouse series of PEC7 models from severe to mild, and we're able to show that even in the milder mouse model, in which there was not elevation of Phytanic acid in several tissues that was detectable, we did have a mild elevation in the cerebellum, and that caused injury to the cerebellar cells. And also in my lab, we have a model for one of the common gene changes in Zellweger spectrum disorder. It's a PEX1 change, and we've been working on retinal gene therapy with that. And with that, I'm collaborating with Joe Hacia, who's here also. And we have some really nice results in the mouse model, and we're really looking at this point to partner in with another company to move this to the clinic. So it's very exciting. Dr. Hacia and I also collaborated on screening cell lines with this common PEX1 mutation for drugs that can recover peroxisome function. And we were successful in finding 2 drugs that we've been working on. And this is a screen that was shifted over to the NIH NCAT Center through a funding by the family organization, the GFPD, and we hope to continue that as a platform that we could screen cell lines from patients with different peroxisomal disorders, and also pick up drugs that might recover peroxisome functions. So I think with that, I'm going to stop and let Dr. Hacia introduce himself.
Hi, I'm Joe Hacia. I'm a geneticist on the faculty of the Keck School of Medicine at the University of Southern California in Los Angeles. So it's nice to be here, and I have a little bit of a long winding path towards being here. But I grew up in New Jersey, and I was a biochemistry major over at Rutgers, which is the State University in New Jersey. And afterwards, I went over to the California Institute of Technology, where I was working with a organic chemist as well as a developmental biologist, and we're working on DNA binding molecules. So I have a little bit of multidisciplinary background, although that means the jack of all trades, master of none potentially, but nevertheless, I could speak the language of a few different fields to some limited extent. And then afterwards, I did a postdoctoral fellowship over at the National Institutes of Health with Francis Collins, who's the departed director of the NIH, but he was the lead of the Human Genome Sequence and the Genome Project. And it was at that time in his lab, I was working on breast cancer diagnostics. I used to be involved in resequencing breast cancer genes, and I developed technologies for that. And Francis got a letter from a scientist named Francis Crick. And for those who know Watson and Crick, they're the co-discoverers of the structure of the double helix, with assistance from Rosalind Franklin, who never got enough of credit for what she did in that project. But nevertheless, Francis Crick was one of my, at least on the scientific idols in terms of his accomplishments. Can't say I know much about who he was as a human being, but I knew Francis Crick was pretty freaking brilliant. So he had this very odd, quirky letter about the need for a gorilla genome project, which was something that was very exciting to me because my mother used to take me to Planet of the Apes movies. And so I was a big fan of animals and the zoo. And I thought this would kind of fit a strange man from New Jersey to work on apes. So I began working on the genetics of human and non-human primates, primarily chimpanzees, bonobos, and gorillas. And I know far more about them than I could actually, anybody would want to know. But actually, it was quite fun working with it at the time. And we were looking at genetic differences across these species. And it was at that time that working with the San Diego Zoo that we started getting skin cells from the animals. And we found that there was an overexpression of Phi-H in human cells relative to non-human primates, at least the limited set that we looked at. And so I was trying to figure out there was a story that we can spin out of this. And the story that I came up with was that there might have been some adaptations in a human lineage to deal with the fact that for historical times, many human populations have had meat in their diet. So it would make sense that you would potentially want to upregulate a detoxifying pathway in the human lineage. Now, whether or not this is a true or not, it was a fun story to spin. And a lot of times academics living in our ivory tower spin stories. But nevertheless, it was a fun one. And I reached out to, reached out to researchers in the field of Phytanic acid biology. And so I first reached out to Ron Wanders in Amsterdam, and then due to some issues about sending cell lines or blood samples from animals across overseas, I then was alerted to Hugo Mosier. So Hugo Mosier is one of the giants in the field of neurogenetics, and although he has passed away several years ago, I was working with his wife Anne, who's to me a living saint, one of my favorite people in the world, along with Paul Watkins, who's also on the board, and that was really through that group over in Baltimore at the Kennedy Krieger Institute and Johns Hopkins. I got to know Nancy, who's a dear friend, even though we disagree on things from time to time. But nevertheless, and she's smiling. So it was really at that time that I started working more on these paroxysmal disorders that Nancy mentioned, and been essentially trying to become a little bit of a hub in terms of connecting people and building communities so that we can bring new technologies and people to the forefront who can be able to bring therapies to these families in need. But it was as a result of, working with Anne and Hugo, it was Anne recommended I go to a ULF meeting. And that's really many years ago where I started meeting with families with these paroxysmal disorders. And eventually I became a member of the board of advisors of the United Leukodystrophy Foundation myself. And that's where I met Kristie. And I think that was in 2019.
Yep. That was right.
Yeah, it was an interesting time. As we were introducing ourselves, I introduced myself as working on a very quirky type of paroxysmal disorders that also had something to do with phytanic acid that nobody in the room would have ever heard of, and there was Kristie, and so we got chatting, and I was very excited about the work that she started and was pioneering with Global DARE, and it's been an honor and privilege to be on this journey with her as we start trying to apply new technologies to this problem. And in this regard, I'm very grateful that we were given a grant from the National Institutes of Health for about $3 million recently to develop animal models of paroxysmal disorders, especially mouse models. And this is going to also encompass Refsum disease. And these animal models really do form the backbone of what is needed to be able to get trials, to get clinical trials going. We have to test the drugs in the preclinical setting, and in the preclinical setting, the mouse is really the major organism of interest where you test, drugs and gene therapies. So we now have a little bit more of a wherewithal to be able to bring these resources to the forefront and hopefully advance research into adult Refsum disease as well as other paroxysmal disorders.
Great. Thanks both Dr. Braverman and Dr. Hacia for your introductions. I've had the pleasure of working with both of you over the last 2.5 years since starting Global DARE, and I really admire the work that both of you do. I think rare disease is one of those thankless jobs, I think, and it seems in research you do a lot of work and a lot of probably late nights, long weekends, trying to make some progress in these rare diseases, and it's a challenge. So I appreciate your efforts in the field. It definitely makes a tremendous difference and gives us all hope. So I think my first question for all of you was around to talk about, what do you think are the promising avenues of research that are potential for Refsum disease? Do you want to start, Dr. Hacia?
Oh, okay. Well, I'll give the researcher perspective, and Nancy's a physician scientist, so I'll defer to her opinions and her expertise on this. There's a few, historically there's been a lot of interest in trying to promote omega oxidation pathways in the liver to clear phytanic acid systemically in patients. So omega oxidation is sort of like you could think of it as a backup pathway for clearing phytanic acid. The alpha oxidation pathway is the one that is dependent upon Phi-H as well as PEC7 functions, but the omega oxidation is independent of that. So although we do not have any small molecule drugs that can be able to stimulate that, say in the liver or other tissues, if we were to have them, that might be an avenue of interest of clearing Phytanic acid. The other area of interest would be to have potential gene therapies where we would introduce a normal copy of the Phy-H gene in the livers of affected individuals, so we can restore the activity of the liver to clear Phytanic acid that's circulating in the body. So the other way, and you can do that for try to reduce the systemic burden, or you can potentially do this in the retina itself. We don't know if that would potentially help or not, but this is a technology that is something that Nancy's group has really pioneered the application of in Zellweger spectrum disorder, which is another peroxisomal disorder caused by mutations in a different set of genes, but primarily PEX1. So there's a lot of hope now that we can be able to at least restore. There's a couple avenues in theory where we can restore Phytanic acid clearance from the body. And as we were putting together a grant with Florian Eichler over at Mass Gen and Joey Scafidi over at the Kennedy Krieger Institute, along with Global DARE, we were also thinking of other ways to try to address some of the neurodegenerative issues that are happening in adult Refsum disease and trying to get at the mechanistic pathways and things that are very, very practical. I tend to think of, as I've grown older and more cynical, I've become more of an engineer in the way that I think, and I'm trying to find ways of trying to get trials out quick, like in the next couple of years instead of waiting 10 or 20 years. And, understanding more about exercise physiology, the rates at which we can, Phytanic acid is liberated from adipose. Why is it that, Phytanic acid itself is so neurotoxic? Is there any way we could be able to prevent it from getting into the tissues of interest, which might be the retina or the cochlea and so on? So there's a lot of, good science to be done, a lot of basic science, and there's also a lot of practical work that can be done that I think can make a difference in the lives of people in adult Refsum disease within the next few years.
I'll hand it over to you now, Dr. Braverman.
Yeah. Okay. Well, yeah. Well, thank you, Joe. I guess one way of thinking about this, which sort of Joe went through all of these possibilities, would be to think about the gene itself, which is Phy-H, so we could try to correct that gene or express the normal gene in the places that we need to. And the other level below that is to replace the RNA that's made from the gene as normal RNA. Again, you have to get to the right places at the right time. And then further downstream, you have the biochemical pathways, which we believe are somehow causing this disease. And so the more we learn about the biology, the more insight we're going to have on different targets that could correct the downstream consequence of this disease, because the gene doesn't work right, so the protein doesn't work right, and in the end we have elevated Phytanic acid that we believe is toxic to tissues. And so those are the downstream pathways. Can you get rid of the Phytanic acid besides dietary therapy? Can you diagnose earlier so that you can keep the Phytanic acid normal throughout life? That might even be something that, and this is a rare disease, but could we get this on the newborn screening in any way? How does Phytanic acid it's probably not there in individuals that early on, so there might, but that would be one way to think about it. The earlier we can diagnose this disease, the better the dietary therapy is going to work, and that's available at the moment. But then there's probably other consequences of this Phytanic acid building up on other pathways and maybe injuring other parts of the cell, like the organelles in the cell. And so once these things are figured out biologically by scientists, then that opens up other pathways for treatment. And this is something that we need to work on as well. And hopefully with the mouse resource that Joe has just talked about. That was a new NIH grant, and hopefully with other grants we can work on this downstream biology of the disease and find other pathways that we might be able to manipulate by drug therapies. There are many different mutations that cause these defects in Phi-H, so it's hard to imagine gene correction because you have too many different changes to correct. And so things like a gene augmentation therapy or an RNA therapy or a drug therapy that could possibly chaperone a misfolded protein, these are all possibilities. And then ways to get rid of the phytanic acid that's building up. And one was omega oxidation that he spoke about. But then I just wanted to add in that the more we understand the toxic consequences of phytanic acid on the tissues, the more we'll find additional pathways that we could treat. And I think it may be, we're thinking in many rare diseases, that one therapy itself may not be curative. And so we maybe need to think about cocktail therapies for many of these disorders. There might be a different cocktail at a different stage of the disease, or at a different age of a person, or at a different expression of the disease in the body. And so there may be multiple therapies. It might be nice to have multiple therapies that we could apply to get better and better effects. So, yeah.
That's very well stated, especially when you're talking, the earlier diagnosis, the better. Ideally, you'd find a way of identifying people through newborn screening, whoever, as Nancy has mentioned, in a newborn, the phytanic acid levels are not going to be elevated. So this is going to be a challenge. But the other point that I should emphasize, which is a point of hope, is that there is an adult Refsum disease mouse model that was first developed by Sasha Fernandez as well as Ron Wanders and colleagues in Amsterdam, and published on, well over about almost like 10 years ago. And those animals that fed phytanic acid or its precursor, phytol, do develop a neurodegenerative course of disease, such as they have troubles with their balance. They certainly have accumulation of phytanic acid in a lot of different tissues. And there's also evidence of peripheral neuropathy. So no one, to my knowledge, has gone in depth to see if there's actually vision loss in these animals. So that's something to think about. But the animal model, as Kristie and Nancy know, is present right now at Johns Hopkins in the laboratory of William Wong. And there's interest from his colleague at the Kennedy Krieger Institute, Joey Scafidi, who's a pediatric neurologist, about potentially taking over that mouse colony. So now that those animals are, in Baltimore, and they're going to be fed phytol, and we're now going to take these animals and start working with them again, so we can be able to understand where phytanic acid accumulates in their body, be able to gain a greater understanding of the neurological degeneration that we're seeing in the animals. How long does it take? What type of diet does it take to make this, to speed up or slow down this accumulation? And then Nancy was kind enough to potentially be looking at the pathology of the eyes of these animals to see if there's any retinal degeneration. Now, if there is, then that opens up the possibility for testing therapies in these animals. But it's never guaranteed that a mouse model is going to recapitulate what you see in a human. It may turn out that we certainly know that there's some liver involvement in the animals and some nervous system involvement, and that's good, especially with the nervous system. We don't know anything about the retina, but if it does, then it opens the door of a possibility of testing some therapies to try to address vision loss in these animals.
Right. Well, thank you both for talking about those avenues. I think lots of good possibilities and a long road ahead. Hopefully, Joe, I always like the quick wins. If we can find things that are, we can do within 2 to 3 years, but also know that some of these things will take much longer potentially too.
I could see this moving fast. We have to have a couple lucky breaks, especially with the eye. May or may not come through, but nevertheless, the animals are there. And, it really does come down, many of these projects, if you're thinking like an engineer, it comes down to funding. And that's why I'm grateful that we put into this Jan Zuckerberg Initiative grant. And even if that does not get funded the first time around, we do have text that can be used to submit grants to other, in National Institutes of Health and other funding institutions. And if Nancy helps pull off the miracle of finding a corporate partner to work with some of the retinal therapies that we've been working as a team on for Zellweger spectrum disorder, that might open up the door really to get these investors also interested in adult Refsum disease, which in my opinion is very well suited for these clinical trials, because you have adults with a degenerative condition that can tell the ophthalmologist or a neurologist exactly how they're feeling that day and exactly the acuity of their vision and how it changed from year to year. And that's something that is very powerful.
That's great. Well, thanks, Joe. There is a question that came in the Q&A. Before I move on to the questions I prepared for you, I'll go to that one first since it's from the audience. So it is, so if we are born with this gene defect, why does the onset of the disease occur at different times across individuals? And what generally triggers this? So which one of you would like to take that first?
Let the physician take care of that, and then I'll back up Nancy.
Dr. Braverman, you're up.
Well, yeah. Well, I have to say that I shouldn't be considered an expert on the treatment of adult Refsum's disease because I'm not. Most of the patients that I'm following and see are in the paroxysmal assembly disorder disease spectrum. But from what I know about these these disorders is that it takes a while for the phytanic acid to accumulate to a level that its toxicity injures tissues, and that becomes a medical problem. And so this would be classified under one of these late onset disorders. And there are many types of late onset disorders that are genetically inherited. For example, Huntington's disease is one of them. And there are familial types of amyotrophic lateral sclerosis, and there are all sorts of spinocerebellar ataxias. And these individuals have gene defects, they're born with them, but the disease doesn't express itself clinically until they're teenagers or early young adults or even middle age. And so this, why is that? And our idea that spans all of these disorders, including adult Refsum, is that something is going on in the cells, and it's taking time for there to be enough of a consequence that there's going to be a tissue damage. And then there's a certain amount of tissue damage that has to occur before it's clinically expressed. So these are some of the reasons why we all tend to think the earlier we diagnose someone, the better, because they'll be at the earliest stage of the disease where there won't have been damage already occurring. So the idea in adult Refsum's disease is that it's this accumulation of phytanic acid, and that's based on the idea that if you can reduce the amount of phytanic acid, you can improve the outcome of the disease. And I think this is the group from the UK that has showed that. And I think one of the physicians is actually here as well on this call. So I think, does that answer the question?
I think so. And I think given the fact that, it's tied to our dietary intake as well. If someone were, say, a vegan from the point of birth, maybe their onset would even be further down the road. So I think it would vary potentially patient to patient based upon their dietary intake, over the course of their life too, I would imagine.
it would be interesting to see if there are any other modifiers that might modify the expression of the disease. There usually always is. Nobody exactly expresses the disease the same, because we have about 30,000 genes that will end up modifying our expressions of disease. And so it could be that somebody has better omega oxidation, for example, and might not express the disease to a later onset. So those are interesting studies. They're called modifier studies. They're not that easy to do, but we have better and better technology that can get a handle on them.
Yeah, that's an excellent point. There's gene and environment interactions. For this particular, disease, you're thinking of the environment, which is this phytanic acid or phytol exposure in the diet, right? But then you might ask ourselves, how do we measure phytanic acid? Well, from the clinical point of view, you would want to get body fluids, something like a blood draw, and that's how you measure phytanic acid. But the key is how much is actually accumulating in the tissues, and we cannot access tissue samples from a human being. You're not going to take a retinal biopsy. You're not going to take brain biopsies or liver biopsies. And as a result, we make an assumption that the amount of circulating phytanic acid in your blood is directly proportional in one way, or at least proportional to the amount of phytanic acid in tissues. And so that could also be a difference between people. Perhaps there's some people that the circulating phytanic acid in the blood just doesn't accumulate in the tissues as much. Now, the only way that we can get at these types of questions is, through an animal model. And it's always been a question to me that, well, we have a lot of phytanic acid in meat products, like say from a cow. Yet at the same time, even though it's chock full of phytanic acid, those animals are not having vision loss with age, nor are they showing any sort of signs of peripheral neuropathy or any sort of disease that one would see in a human being. And why is that? That has to do with something about sequestration of phytanic acid in different tissues in the body, or there are adaptations metabolically in these animals where phytanic acid accumulation would be relatively non-toxic to them, but in a human being actually causes some sort of pathology through an unknown route. And even though there's hypotheses or guesses about why phytanic acid is neurotoxic, we don't know precisely why it is.
We also don't know if it has, if I can just add in here, we also don't know if it has a biological role itself. Maybe a certain amount of phytanic acid is important. It is known to be able to activate certain types of receptors in the body called peroxisome proliferator activator receptors. So I've always wondered whether or not it is completely some sort of artifact compound that is always toxic, or whether it does at some level have an important role.
Right. It's almost impossible to get at that particular problem because unless it's in a lab animal, everybody, even on a vegan diet, is going to be exposed to these phytol esters in plant products. So there's always going to be trace amounts. It's hard to get it down to zero. Even in animal chow, you'd have to get like a completely synthetic form of the animal chow that has absolutely no phytanic or phytol in it. Maybe you can get at it. But, and that's just like an area of biology where you end up having to do with trace exposures to, like xenobiotics, that we would call it, or, exogenous types of chemicals from our diet. And, trying to decipher whether or not, 1 part per billion or trillion is, important for you or is like trivial, etc. So it is a good point. And just one of these questions that sometimes we can't readily answer with the technology available to us.
Another interesting mode of therapy, I just thought when you were talking about a diet, is there's a company in Boston that's looking at oral drugs or biologically active oral compounds that will actually break down the toxic compound as you eat it. And so they have this available for patients with PKU or phenylketonuria, where they take a bioactive substance that breaks down the phenylalanine as they're eating it. So it doesn't get into their bloodstream, or much more reduced amounts gets into their bloodstream. So this would be a fabulous idea there. Can you find some sort of probiotic, some sort of organism that you could digest and you could break down the phytanic acid before you actually absorb it in your gut? That would be another possibility.
Mm-hmm. There's a lot of things to think about.
Just sometimes just by having these conversations, right? I just flow, and you never know what you might come up with as ideas. I think it's when I think about other rare diseases and their application and what they're doing, as you say, you mentioned a couple of other metabolic disorders that there's some sort of therapy. I think that that's why it's so important to communicate across rare diseases, right? Because there are many things that could be applicable, things that you're doing on other peroxisomal disorders, as Joe said, on the PEX1 mouse for retinal therapy could be very applicable for Refsum. The idea that you could eat something that could break down the phytanic acid, I could have some cheese, that would be great, Those little things, but yeah, one of the other questions I had for you. So we had a couple, Dr. Fred Vaz and Dr. Michael Wengler, both did did some pre-recorded videos on metabolomic and lipidomic studies and how those could be important to Refsum's disease. So I was wondering if either of you have had experience where those things have provided some valuable insights to other rare diseases in peroxisomal disorders or elsewhere, where doing those studies made a difference in finding other reasons, besides, say, for us, a biomarker of phytanic acid. Maybe there is something else going on, as you said, that is causing some of the progression and disease. You want to start?
I think Nancy could take over because she's worked with Mike, and I was just peripheral to it, but looking for these metabolomic profiling in people with Zellweger spectrum disorder, as well as rhizomelic chondrodysplasia punctata, primarily type 1.
Yeah. So the idea with profiling body metabolites and body lipids, and body proteins. You could do proteomics, you could do metabolomics, you could do lipidomics. And the idea is to get a global view of all of these compounds and how they're altered in patients with the disorder versus a patient without the disorder. And the end goal is to try to see if you can figure out a novel pathophysiology, which we just talked about is always interesting, because that always opens up novel therapy, because it's something you didn't think of before that could be causing the disease. And you can usually at that point, or hopefully at that point, figure out a way to correct that, but you couldn't before because you didn't even know it existed. So this is one reason we do these types of studies. The other reason we do it is to look for biomarkers that we can use to prognosticate how the disease is evolving, or prognosticate how the disease is responding to a therapy, or use as a diagnostic marker. So in this case, maybe one of these studies could come up with something that might be present in adult Refsum's disease in infancy and could be added to newborn screening. I'm just giving an example. That wasn't the reason that we did the studies, but these are the things that can come up. This would be called a biomarker, a diagnostic biomarker. So we look at these types of omic studies or global studies of everything that's happening to chemical compounds in the blood. That's the metabolomics, or lipid compounds in the blood is the lipidomics, or all of the protein signatures in the blood, which is proteomics, to really look for new biology about the progression of the disease, and to find new biomarkers to assign for diagnosis or prognosis or response to therapy. And specifically for peroxisome disorders, one of the markers that came up that Mike Wengler published was sphingomyelin that was decreased for carrying certain fatty acids on it. This is a membrane phospholipid, but most likely increased for carrying the very long chain fatty acids because they're accumulating. We know that they're sticking onto membrane lipids, and now we have a marker. We have this membrane lipid called a sphingomyelin that's now sort of a net for these very long chain fatty acids, and we could start looking at that as a diagnostic or prognostic marker or influencing the disease severity.
And I think that's a good point about how, for many of these paroxysmal disorders, from Zellweger spectrum disorder, which is principally a problem with the assembly of peroxisomes, and so you can imagine just about every function in the peroxisome is now impaired to a certain degree. It could be completely, in which case the prognosis is very poor, or it could be partially, in which case you have a better prognosis. Or you can go to adult Refsum disease, where it's a single enzyme deficiency, as we describe it, just one particular pathway in the peroxisome. Or it could be adrenal leukodystrophy, which they focus on at the United Leukodystrophy Foundation, which has to do with a defect in the turnover of very long chain fatty acids. Nobody really knows the pathomechanisms of the disease completely, but that's what's going on there. And by combining data from the metabolomic profiling of all these different groups of patients, we actually get a better picture about what's going on with the pathomechanisms of each one, and a better idea of what's going on with peroxisome biology. So there's a community spirit there, and it's we appreciate when people participate in these studies, and I hope they can see that there's the potential to you could do good within your own community as well as across many others.
So just to touch as one of our final questions, because we're coming up on time, is, to touching a little bit on that, Joe, how can patients best participate in research?
Well, I'm gonna talk to Nancy. Nancy's the physician here, and I can be able to back her up on this. No, and I do it with all due respect. I'm not a physician, so I shouldn't be the one, talking principally about medical issues, even though it's near and dear to my heart. But I do like to, kind of add on when Nancy says something eloquent, which she has been doing.
Thank you. I think patient advocacy groups are hugely powerful. So I've been in this field, since the early 1990s, and the DARE Foundation wasn't here, the Global Foundation for Paroxysmal Disorders wasn't here, Zellweger UK wasn't here, PBD Canada wasn't here, and with the formation of these organizations, bringing families together and advocating for their diseases has hugely expedited this course towards therapy, because the family organizations are really the ones that are getting people aware of these disorders. They're educating, they're advocating for the best management for their children and their family members, and each individual patient is advocating for themselves in this stronger organization that can then advocate together. And so I think this is very, very empowering in a rare disease field to have a family organization that advocates for improving care in these disorders, and then also for fundraising, as Joe had mentioned, is that the issue with rare disease is that there probably is a cure, or there's a cocktail, as I mentioned before, or there's something we can do to improve the course of these disorders, but it's just when. And the more funds that we can put into it, the faster that we're going to find it, because we all believe it's there. We just have to figure it out. And if we have the funds to figure that out, we could get more therapies to patients, and of course more individuals and more scientists involved in working on these disorders, and more physicians involved in taking care of patients. Everybody together, I think, has a role to play, and then together, we're much a stronger group in getting the things that we want. So aside from that, then you have these natural history studies, which are of huge benefit, and because they not only help map the progression of the disease in individual patients, and that helps us tailor management and improve patients' care from what we have, existing resources we have today. But the natural history study also gives us accurate endpoints for therapies when we can start to provide them. Because if we don't know the course of the disease, then we don't know if the therapy is going to work. And that's really, really important. So these natural history studies can help us develop clinical endpoints that will be strong, that will be accurate, that will be solid. That's what patients want to see. You know, they don't want a clinical endpoint that they couldn't care less about. They want to know that, you know, something functional is happening in their disease. And so we can't establish those clinical endpoints accurately unless we get a really good idea of the natural history of the progression of the disorder in different individuals, so we get a grasp on this variation that's going to be in any disease, but can be particularly large in some disorders. So I think that's how patient helps. And then patients help when clinical trials come along, you know, and we don't know if the therapy is going to work, and that's why we're trialing it. And it's really important for patients to get involved and to be in it, even though there's going to have to be a control group, because the only way we're going to know if the therapy is working is if we can compare it to someone who didn't get it. You know, the idea would be to design the trial so there's less people in the control group and more people who are actually getting the therapy. And the trial is designed to be only over a short period of time, relatively short period of time, so that we will know if it works or not, and then we could get it approved or get it extended where everyone's on the drug. And this has been going on for other rare diseases. The type of trials that we're planning has changed, but it's really important to have patients be interested in going into these trials, even if they don't know if it's going to work or not, because we won't find out if we trial it.
And I was especially interested, for what Global DARE has done. You know, the impact it had on me was, you know, the awareness of just how many people are affected with adult Refsum disease. You know, I live in my own little ivory tower where my contacts are obviously a number of other scientists in the field, but mainly in the world of Zellweger spectrum disorder and, you know, reading the literature. But I thought there was only maybe about like 5 people in the world who might have had adult Refsum disease, if you were to ask me like about, you know, 4 or 5 years ago. And, you know, then once I met Kristie, and then you actually, makes a big difference when you actually meet a person and you get a personal connection. And, you know, as a result, I've seen the scientific community grow. We have these wonderful meetings of our medical and scientific advisory board. We've had a little bit of a renaissance in peroxisome biology over at the Kennedy Krieger Institute in Baltimore. There's a couple other people coming on board right now, and that makes a big difference. And as Nancy mentioned, and again, this is the part that, you know, always concerns me as a pragmatist and also as an idealist, is that idealistically, when there's technologies that can help people in the real world, why have we developed gene therapy? Why do we develop all these drug screening technologies if, in fact, we can't help the people that we're attempting, that, you know, are paying the taxes, who are actually in need of these advances? So some of the gene therapies that I've seen that have advanced in the clinic have advanced. The most rapid ones are foundations that have actually flipped the bill pretty much by themselves. And so then you'd ask the question of equity. You know, is it only the folks that could actually raise $7 to $10 million? Are they the only ones that can potentially have access to a given therapy? There's other avenues, as Nancy mentioned. You could get biotech support, and that's possible. But it still is not an easy task to get a company interested in an ultra-rare disease. It is certainly possible, but it might take many, many years. Grants, you can say, well, maybe that's 10 or 20 years. And if I was the person affected by a disease or my loved one, 10 to 20 years is not acceptable. I want something faster than that. And that's where foundations can come in, and they can help spur it. They don't necessarily have to raise all the, you know, $7 to $10 million. Coming from a working class family, I recognize it's fairly obnoxious to say. I know I couldn't even be able to raise, you know, a few $100s if I were to, you know, put out a Facebook, you know, like a fundraising relative to actually do something where I had to think of millions. But even, the amounts that Global Dare has actually put together to date to be able to help spur the mouse model work and, thinking about supporting some of the clinical centers of excellence in Boston, which is going to help a lot of different communities. That's inspiring, and it is going to make a difference.
Thanks, Joe and Nancy. So I think I really appreciate the both of you joining today and answering and providing your viewpoints in on the avenues of therapy, how various avenues of research could help us from an adult Refsum perspective, and paroxysmal disorders in general, as well as how us as patients can help in this journey. So do you have any parting comments you'd like to say before we wrap up? Otherwise, I'll close out.
Well, I just want to thank you, Kristie, and for putting together DareFest. I want to thank Nancy for coming in and joining in. She's, one of the best paroxysmal disorders doctors in the world, and a very kind and compassionate human being. And this has been wonderful, and it's been good for my spirits working with you, Kristie. I've really just enjoyed the opportunity of being a patient advocate and trying to do what I can to build a community of researchers. It's all, as Nancy mentioned, it's a team. It's not just one lab. There's a lot of different areas of expertise that are needed to be able to put a product out. But we're doing the right thing. I am confident that there is a solution to improve the lives of people with adult Refsum disease. And I feel it's a moral imperative for myself, as long as I'm physically able to do so, to dedicate myself to make this happen.
Great. Thank you both. I really do appreciate both of you, your time and your ongoing engagement with Global Dare and helping us towards our mission of improving the lives of people with adult Refsum disease, as well as other paroxysmal disorders. As I think I've said earlier, we've got quite a few different paroxysmal disorders that connect with us just because of our low Phytanic acid diet. So I think the more we can connect and partner, the better. So I want to thank all the audience for joining and listening and engaging with us today on this important topic. So that ends today's panel, and I hope everyone's enjoyed DareFest. I know I've enjoyed this first annual Refsum Awareness Week. We've got more activities for the rest of the week planned, and it should be great. So thank you very much.
Thank you.